Today, we're talking about a technique that truly changed the game in molecular biology, Southern blotting. It's all about finding that one specific gene, a genetic needle in a massive haystack. So, to really get why this was so important, you have to understand the absolutely massive problem scientists were facing back in the early 1970s. I mean, think about it for a second. The human genome has over 3 billion letters in its code. How on earth do you pinpoint one tiny specific sequence in all of that noise? It seemed almost impossible. And this analogy, it's just perfect. See, to even start to analyze DNA, you first have to chop it up into millions of little pieces. So you're not just looking for a sentence in a giant library, you're trying to find it in the pile of confetti that's left after that whole library went through a shredder. And this is the brilliant solution, a technique that seems almost like magic, developed by a British biologist named Edwin Southern. It lets scientists pull that one specific piece of confetti, that one gene, right out of the mess so they can actually see it. You know, the story behind this invention isn't just about the science itself, it's about the scientist. And the inspiration for his breakthrough idea? Yeah, it's probably not what you're thinking. Southern's idea didn't just appear out of nowhere. It was this incredible synthesis of technologies that were brand new at the time. Scientists had just figured out how to use things called restriction enzymes to cut DNA and how to use gel electrophoresis to sort the pieces by size. Southern's genius was seeing how these new tools could all work together. And this is the best part. His motivation was basically efficiency. He later joked that laziness was the real father of his invention. The old way of doing things meant physically slicing up gels into hundreds of tiny pieces. It was slow and incredibly tedious. He just knew there had to be a better way. And this just says everything about the scientific spirit of the time. Before he even published his paper in 1975, Southern was just giving the idea away. He drew this diagram on a scrap of paper for another scientist who took it back to the US. The technique spread by word of mouth. It was pure open science. Okay, so how does this better way actually work? Let's walk through the blueprint step by step. The whole thing really boils down to these 6 key steps. We're going to go through each one to see how you get from a huge, messy sample of DNA to a single, clear result. All right, step 1, you take your DNA and use these little molecular scissors. They're called restriction enzymes to chop it all up. Then you put those fragments into a gel. An electric current pulls the DNA through it, and since smaller pieces move faster and farther, everything gets sorted by size. This is the shredding and sorting part of our library analogy. Now here's where the real magic happens. This is the blot itself. The gel is super flimsy and hard to work with, so you lay a tough membrane right on top of it. Then you stack paper towels on that, which creates this capillary action, kind of like how a paper towel soaks up a spill. It pulls a buffer solution right up through the gel, carrying the DNA fragments with it and sticking them to the membrane. What you get is a perfect, stable copy of the DNA pattern. Okay, now it's time to find our gene. We create something called a probe, which is a small piece of DNA that's designed to be the exact opposite, the perfect complementary match to the sequence we're hunting for. We wash this probe over the membrane, and thanks to the rules of DNA base pairing, it'll only stick to its one true match, completely ignoring the millions of other fragments. It's like a highly trained search dog. So we've done it. The probe has found our needle in the haystack. It's latched on, but how do we actually see it? This is the final step. Well, that's where the label on our probe comes in. Back in the day, these probes were made radioactive. So after you wash away any extra probes, you'd put the membrane against a piece of X-ray film. Wherever the radioactive probe was stuck, it would expose the film, leaving a dark band. These days, we can also use fluorescent tags that glow under certain light. And there you have it. This is the payoff, the eureka moment. Each one of these dark bands is our target DNA fragment, the very thing we were looking for. We found our sentence in the shredded library. And this simple pattern can tell a scientist all sorts of things, like the size of the gene or even how many copies of it there are. For decades after its invention, the Southern blot was an absolute cornerstone of molecular biology. Its impact was, and honestly still is, just immense. And the applications? Oh, they were total game changers. Southern blotting led directly to the first DNA fingerprints used in crime scenes. It became a critical tool for diagnosing genetic diseases, for understanding how genes get messed up in cancer, and for the massive project of mapping the human genome. It was absolutely revolutionary. And here's a little bit of science humor for you. The name Southern blot became so iconic that when scientists developed similar methods for other molecules, they couldn't resist a good pun. The technique for detecting RNA was named the Northern blot, and the one for proteins, you guessed it, the Western blot. Which kind of brings us to a final, pretty big question. Edwin Southern shared his method freely before he even published it. He never filed for a patent. He just believed that scientific tools should be open for everyone. When you look at the world of science and intellectual property today, you have to wonder, right? Could a breakthrough this huge ever be shared so openly again? It's definitely something to think about.